Hi. All right, in the second part of this chapter, let's talk about enzymes as biological catalysts. So an enzyme or a catalyst makes an energetically favorable reaction go faster. They cannot make non-spontaneous reactions occur without adding energy, but they can take any spontaneous reaction and make it happen faster. Because as we saw with the sugar example, if I have a bowl of sugar at room temperature, even though converting to carbon dioxide is a spontaneous reaction, that doesn't mean it's just going to happen right now. So enzymes at biological temperatures help reactions go faster. Enzymes, just like any other catalyst, are not changed during the reaction, and they can be reused many thousands of times. If the reaction is non-spontaneous, they can add ATP to help destabilize bonds and make the reactions happen. One place where we encounter catalysts is in our cars. Our cars have catalytic converters that use metals to help get rid of nitrites that are a product of burning fuel, and that contributes to smog. So having a catalytic converter is one of the reasons that the city of Chicago is no longer covered by a brown blanket like it was in the 1970s when catalytic converters weren't found in very many cars yet. And interestingly, in chemistry, metals are used all the time as catalysts. Enzymes also often have a metal ion as part of their structure and require that for function. This is the reason why we have so many minerals. They usually are called on a vitamin bottle. There are so many essential minerals. Many of them, this is their function. They function as part of a catalyst, but in an enzyme, not by themselves, because then they are controllable. If you just have metal ions acting as catalysts loose in cells, there's no way to control that. But if it's attached to an enzyme, we can turn it on and off, and we'll talk about that too. Okay, so what catalysts do is reduce what we call the activation energy of a reaction. So here we have a non-spontaneous reaction. So an enzyme cannot help this reaction unless it adds energy. But these two represent spontaneous reactions because they're downhill. Energy is on the y-axis here. The energy of the product is lower than the reactant. It's downhill. But like our glucose at room temperature, why isn't it reacting? Well, because there's activation energy. This is the energy to destabilize the bonds. So covalent bonds are quite stable, and to get it to change from a glucose molecule into 6 carbon dioxide molecules, you have to break the bonds. And you can do it in several ways. You could just heat it up. So you could take your pan full of dry sugar and heat it on the stove until it combusts. Our cells, unfortunately, don't have that option. Our cells cannot heat up to get to the activation energy for reactions. Instead, what enzymes do is change the shape of the bonds to destabilize them. And they can do that in a couple of different ways. One way would be just to move those two atoms further apart. So remember that in a bond, they're sharing electrons. The nuclei of the two atoms have to be a particular distance apart. It's very distance sensitive. And if you use an enzyme to pull those nuclei a little further apart, that destabilizes the bond. And you can also destabilize by adding a phosphate, as we saw in our example. If you add a phosphate, that pulls away some of the electrons from the bond and also destabilizes the bond. So what that does, when you destabilize, you lower the activation energy. And that means that even though we haven't changed the delta G here, the change in energy is still the same. But now this spontaneous reaction is going to occur at a much lower temperature. It's going to occur at the temperature the cell is. And that's what enzymes do. they allow these reactions to occur quickly without adding any heat. So let's take an example here. So sucrose is a disaccharide, which we learned before, and it's very stable. Even if you dissolve it in water, it's pretty stable. So if you take sucrose and just dissolve it in water and leave it at room temperature, it's going to very slowly break down into glucose and fructose. It probably would take, at room temperature, it might take 6 months to break down into glucose and fructose, but it will if it's dissolved in water. We can make this reaction happen in a couple of minutes, however, by adding an enzyme, sucrase. So when we don't need any energy, because this is a downhill reaction, sucrase isn't adding any ATP, we don't need any of that. But what it is doing is destabilizing the bonds. It's destabilizing the bond between one of the oxygens and one of the sugar molecules. So our covalent bond is made of electrons. And the enzyme, all that enzyme has to do is just pull them a little bit further apart. Just bend that molecule a little bit. And then it will go, because it's a spontaneous reaction. So here's our reactants again. Here's our products. And the activation energy is the energy needed to destabilize those bonds. And if we stretch the bonds in some way by moving the atoms further apart, by adding a phosphate nearby that's going to draw off some of that electron cloud, that's going to lower this activation energy. It does not change the change in free energy. It does not change the delta G. It is not adding any energy. Enzymes do not add energy to a spontaneous reaction. It's not needed. They can add energy if it's needed, if it's an energetic reaction. But for a normal reaction, like for sucrase, helping this reaction to go faster, doesn't need to add any energy because it's already a spontaneous reaction. So with our enzyme, we just lower the activation energy enough so that it's going to occur at cellular temperature, so that this spontaneous reaction that without an enzyme would occur, but it would be very, very slow at cellular temperatures. We add an enzyme, now it will occur extremely rapidly, and we did not add any energy. We don't need to. It's a spontaneous reaction. Enzymes often change their shape. This is called induced fit. So when the target molecule binds to the active site of the enzyme changes shape in order to destabilize the bonds. It changes its own shape and it changes the shape of the reactant so that the targeted bond is weakened by that shape change. Sometimes all an enzyme does is bring two molecules really close together that are going to react, but they're rare in the cytoplasm and they're hardly ever going to bump into each other. And it provides a binding site to get both molecules right next to each other so that they can react. Enzymes are used over and over again. The enzyme is not changed by the reaction. If it is lowering the activation energy and not just bringing them together, which is what most enzymes are doing. This has to be a spontaneous reaction. Going from our substrate to the products has to be spontaneous in order for the enzyme to do it. If it's, for most reactions, it is not adding energy to make this happen. If it's an endergonic reaction, then there's going to have to be multiple steps where ATP is going to be added to the reaction to make the reaction occur, to turn it into a spontaneous reaction. So where our substrate binds, that is called the active site. Here's two cellular enzymes that are present in most of your cells. A particular enzyme usually only binds to one substrate. And its particular shape, its active site, only allows one thing to bind, and sometimes it's very small, sometimes it's very big. So catalase is one of the enzymes that is very common in all eukaryotic cells, and also some bacteria as well have catalase. Catalase gets rid of a waste product from metabolism, hydrogen peroxide. H2O2 there is hydrogen peroxide. And it's a waste product of oxidative metabolism. So our cells have catalase to turn this into water and oxygen. But that's all it does. Catalase can't do anything else. Its active site can only bind hydrogen peroxide, and breaking down hydrogen peroxide into water and oxygen is a spontaneous reaction. It's downhill. It happens at cellular temperatures very slowly. That's why if you buy hydrogen peroxide, it's in a brown bottle. It helps keep light from hitting it, which will also make this reaction occur more quickly. DNA polymerase, and here we see something, notice this, that this enzyme has ASE. DNA polymerase, another enzyme, ASE. That ending tells you that this molecule is an enzyme. Not every enzyme has ASE at the end, but these two do. So, and often the name of the enzyme gives you a pretty good idea of what it does. So catalase doesn't, because this is a really old name that's been around for 100 years. If we were going to, another alternate name of this enzyme that tells us what it does would be hydrogen peroxidase. DNA polymerase polymerizes nucleotides into DNA. It makes the polymer. That's why it's called polymerase. So DNA polymerase makes DNA. So this green blob here, that's DNA polymerase. And down at the bottom here, we can see a double helix. And up at the top here is only a single DNA backbone. And the DNA polymerase is moving along, putting in the second or complementary nucleotide. So we're going to talk more about DNA polymerase at the end of, towards the end of the semester. So very often, if you don't know what an enzyme does, look at the name, because it very often gives you a very good idea in the name exactly what that enzyme does. So what enzyme makes RNA from RNA nucleotides? RNA polymerase. Enzymes are, because they're folded proteins, the shape of the protein has to be correct for it to work. So anything that's going to denature an enzyme is going to affect enzyme function. So remember when we talked about ways to denature proteins, if you use high salt, if you change the temperature, especially high temperature, if you change the pH, especially making it acidic, those things tend to denature protein. So anything that's going to denature protein is going to affect the function of enzymes. And every enzyme has an optimal temperature, an optimal pH, optimal salt levels at which it works. That's the conditions under which it is folded correctly. So here's an example here for temperature. So human body temperature is about 37 degrees C. So we would expect human enzymes to all have an optimal functional temperature right around human body temperature, because why would we have an enzyme that doesn't function at human body temperature? But organisms that live at other temperatures, like heat-tolerant bacteria or Archaea, they have enzymes with other optimal temperatures at the environment that they live. In fact, we use one of these heat-tolerant DNA polymerases for PCR. So we would expect an organism that lives at 70 to 80 degrees C to have enzymes that is around their optimal temperature. pH is also important. In fact, we have two enzymes in our digestive system, one in our stomach, pepsin, and one in our small intestine, trypsin, that have similar functions. They both digest protein and break it down into, pepsin breaks down large proteins into small peptides. That's why it's called pepsin. And trypsin breaks down these small peptides into individual amino acids so that they can be absorbed. So they have similar functions. They're breaking peptide bonds. That's what they both do. And they have completely different optimal pHs. So our stomachs are very acidic and have a pH of around 2. That is where the optional pH of pepsin is found, around pH of 2. Trypsin, on the other hand, because it's in the small intestine is slightly alkaline, so it has an optimal pH of around 8. And if you try to switch them, if you put trypsin in an acidic environment or pepsin in an alkaline environment, they don't function at all. They have no functionality because their shape is going to be wrong, because pH affects hydrogen bonding, and hydrogen bonding is one of the things that folds up proteins correctly. So if you mess with the hydrogen bonding, you're going to change the shape of the protein and destroy its function. One of the enzymes that we would have been using had we been doing our in-person labs would be Taq polymerase. Taq polymerase is a DNA polymerase that was discovered in an Archaea that lives in Yellowstone National Park. And the species name is Thermus aquaticus. That's where we get the TAQ. The T, so it's like T aquaticus, but it's just the first two. And Thermus aquaticus lives at, its optimal temperature is between 70 and 80 degrees Celsius. It can survive being boiled, so it lives around these geysers that are found in Yellowstone National Park and boiling lakes. So they can tolerate being splashed with 100-degree water multiple times a day. So their DNA polymerase, when it's heated up to 100 degrees, it denatures. But when it cools back down, it goes back to its working form and will work again, which is not something that most organisms' enzymes are capable of, that is recovering from denaturing. So it's been very useful. And the PCR, anytime you hear PCR, they're using well, there's another polymerase they use now too from a different organism, but Taq polymerase is still the one that's mostly used, and it allows for amplification of small segments of DNA or RNA, so we can make millions of copies of them so we can study them. Often enzymes are embedded in membranes and localized together so that they can work in biochemical pathways. And we will see this in mitochondria and also in the chloroplast, is that the membranes are used as a way to localize enzymes together. Now, enzymes have to be controlled. We can't have all the enzymes in our cell working all the time. Instead, cells need a way to turn them off and turn them back on. And that's really what inhibition means. So here's another fancy word for a simple concept. Inhibit just means stop. So if an enzyme is being inhibited, it means it's not working right now. It's temporarily being stopped from working. And there are two basic types of inhibitors of enzymes: competitive and non-competitive inhibitors. So a competitive inhibitor binds to the active site. It binds where the substrate would bind and just physically blocks it from getting into the active site. It competes with the substrate for the active site. That's why we call it a competitive inhibitor. Non-competitive inhibitors bind somewhere else. They bind somewhere else on the protein, change the shape of the active site so that the substrate can't bind anymore. And these are reversible. So both a competitive inhibitor and a non-competitive inhibitor can bind to inhibit the enzyme and then release to allow the enzyme to be used again. Wouldn't be much good as inhibition if it permanently destroyed the function of the enzyme. So it's temporary. It temporarily stops the function of the enzyme. And I have linked to a video here that you can watch. Sometimes it's nice to see things visually, but it's just a little short video on competitive and non-competitive inhibition. There are competitive inhibitors in the nervous system as well. In fact, that's how many drugs work, is that they're competitive inhibitors of some natural substance in the body, and caffeine is one of those. It's a competitive inhibitor, not of an enzyme, but of a receptor in our nervous system. So adenosine, which is ATP without any phosphates, can see it's just got the ribose and the adenine. This is very abundant in the extracellular space in our nervous system. And in fact, we have receptors on all our nerves and on our muscle cells too that can bind adenosine. And when adenosine binds to the receptor, it tells that nerve or that muscle cell, don't have an action potential, don't fire. Calming. Adenosine is calming to the nervous system. In fact, it's what makes you sleepy when you haven't had enough rest. So as adenosine, the more hours you're awake, adenosine builds up in the nervous system, it builds up in your brain, and it makes you sleepier and sleepier until sleep is it's irresistible and you fall asleep. Caffeine can bind to the adenosine receptor, but it doesn't calm the nervous system. It just takes up space and it just keeps the adenosine out of that receptor. So that's why caffeine wakes you up, because it keeps the adenosine from binding to its receptor and calming the nervous system and making you sleepy. It has a limit though. Eventually there isn't enough caffeine and you must sleep. All of these inhibitions are reversibly controlled, and very often the end product, or something close to the end product, is actually the inhibitor. So here's another synthesis reaction. Isoleucine is another amino acid that we need to make protein, and threonine it's another amino acid that's not used for protein, but is like an intermediate. And we can synthesize isoleucine from threonine, and isoleucine is actually the inhibitor. So, and all of the enzymes are in a biosynthetic pathway, and I see here there's actually 5 enzymes that are needed to go from threonine to isoleucine. We don't have to regulate every enzyme in the pathway. We only need to regulate the first one, because if this first enzyme is not working, there won't be any intermediates for any of the other enzymes, and they'll just sit around doing nothing, which is what we would want, right, if we wanted to stop. So if there's lots of isoleucine around, isoleucine actually can bind to this first enzyme, threonine deaminase, and or aminase, not animase. Deaminase, it's taking the amine off of the threonine. So it actually binds to it and inhibits this enzyme from working. And then as isoleucine gets used up in protein synthesis and becomes more rare in the cytoplasm, it will be released from this enzyme, and now threonine can bind and the synthesis will continue again. So this is called negative feedback, when the feedback causes a decrease in something. That's negative feedback. We will see this kind of regulation and localization of enzymes when we do respiration, which is the next chapter that we're going to. In mitochondria, some of the reactions are localized to the matrix, which is what we call the cytoplasm of the ancient bacteria. And some of the reactions are localized to the membrane. And in fact, that membrane is used as a source of potential energy. That inner folded membrane of the mitochondria, there will be a buildup of hydrogen ions on one side of the membrane, and those are going to be used to synthesize ATP. So we're going to tie in a lot of this in the next chapter. So again, energy in biology, we're interested in the ability to do work. The first law, energy is not created or destroyed. It is only transformed from one form to another. So in biology, the kind of energy transfer we're most interested in is energy of sunlight being converted into the potential energy in chemical bonds and the potential energy of ion gradients, and that potential energy being used to do cellular work. The second law, entropy increases in a closed system. Yes, and that also means that if you want to decrease entropy, like build things up from smaller molecules, make DNA from nucleotides, that means you have to add energy. If you're trying to decrease entropy, You need to input energy into the system. So spontaneous reactions are energetically favored. They have a negative delta G. They are downhill. They release energy. Reactions with a positive delta G require energy to be input into those reactions in order for them to go. And enzymes are how all these reactions are taking place. Almost every chemical reaction that's taking place in your cells needs an enzyme to make it go. Enzymes work the same way catalysts work, by reducing the activation energy so that you don't have to change the temperature to make the reaction go, because cells cannot heat up to lower the or to reach the activation energy. Instead, they lower the activation energy by destabilizing bonds. Competitive versus non-competitive inhibition of enzyme. Competitive inhibition means that our inhibitor is going to bind right to the active site of that enzyme. ATP is the energy that's used by cells, mostly. Sometimes GTP is used. And enzyme activity is affected by temperature and pH, and also salt concentration, because those things affect the shape of proteins. And an enzyme is a folded protein. It has to be the right shape in order to work. Okay, that's the end of metabolism, energy, and enzymes. In the next chapter, we're going to talk about respiration and mitochondria.